We know that immune response can be generated against sperms, and since the spermatogenesis it begins during puberty, normally the tight Sertoli cell junctions, and that are the tight junctions between Sertoli cells, they provide the testes barrier from blood. So the blood is not in direct connection with the sperm cells. So sometimes these barriers, they get avoided, they get loose. So during torsion, testicular torsion, vasotomy, testicular trauma, testicular surgeries, these barriers, they get weak. So sperm get exposed to the immune system, and then the antibodies, they are formed, because we know the sperms, they have the specific type of proteins that are not present in other part of the body, and these cells, they are also haploid cells that are not present in other parts of the body, so they appear to be different cells to the immune system, so immune system attack it. So anti-sperm antibodies, they get attached to the sperms at various sites. They could attack to head, they could attack to the tail, they could attack to the neck, anywhere on the sperms. So they could attack many sperms together, so cause agglutination. So the sperms, they get trapped, they are not free to swim to ovum to fertilize it. There could be sperm antibodies that are without sperm agglutination. They are only lining the heads of the sperm or tail or the neck of the sperm. Or they could be in agglutination. That agglutination means the sperms, they get trapped, more than one sperm, they are attached to single antibodies. So these antibodies, they could be IgA and IgG. We know the IgA, it is present in the mucus of the body, and it is normally present. And when it get exposed to the sperms, it get attached to sperm. And IgG is the antibodies that is formed for long-term infections. So when the sperms, they get exposed for longer time, they develop IgG. So if we determine antibodies in semen, and if less than 50% of the sperms are bound with the antibodies, then we say the semen is functioning normal. So it is not having sufficient anti-sperm antibodies that can cause pathology. There are two types of tests for anti-sperm antibodies. There could be a direct test that tests the presence of antibodies on spermatozoa, or there could be indirect tests that determine anti-sperm antibodies in the sperm free fluid, that is the seminal plasma or blood serum, and or the cervical mucus. So these two tests, that is direct test and indirect test, they are being used. So these tests are MAR test, that is mixed anti-globulin reaction test, or it could be Immunobleed test. These are the two tests that are being used. They could be direct or they could be indirect. Direct mean they will determine antibodies on spermatozoa, and indirect mean they will determine in any fluid of the reproductive tract antibodies that are not attached to sperm, that are freely swimming in fluid. So MAR test, it is used, that is mixed anti-globulin reaction test. It is used for fresh semen without washing it, while Immunobleed test is used on the semen that is washed for spermatozoa. So spermatozoa, they are separated from other antigens. We know the sperms, they have anti-sperm antibodies IgA or IgG. In order to determine these antibodies, we have we need the fresh semen, and then we need the RBCs are now they are replaced with latex particles. So we need latex particle, they are like plastic particle. So these particles, they are bound with, they are coated with anti-sperm antibodies, either IgG or it could be IgA. So in case of sperms, they are also, if they have antibodies, they would be IgG or IgA antibodies. So same antibodies, they are bound with RBCs, or they are bound with the latex particles. And then we need another thing, that is the uh, anti-sperm antibodies, binding antibodies. So these antibodies, that are the antibodies that can bind with these anti-sperm antibodies. So these antibodies, they have their domains that can bind with more than two antibodies. So we first take the fresh semen, and then we add the latex particle that are also bound with the anti-sperm antibodies, and then we add, on the same slide, we take for example 5 microliter fresh semen, 5 microliter of these latex particle that are coated with anti-sperm antibodies, and then we add these linker antibodies. These antibodies, they are also called as bridging antibodies. So we add these antibodies on 5 microliter on the same drop of the uh, semen and the latex particles, and then we mix it and put a slide and see these linker antibodies, they can bind with more than one anti-sperm antibody. So one of their domain can bind with antibodies present on the latex, and the second could bind with the antibodies present on the sperm. If the sperms, they have antibodies, then we know the particles, they definitely have antibodies, but sperms, we are suspecting either they have antibodies or they don't have antibodies. So if they have antibodies, then these linker antibodies, they would link, or bridging antibodies, they would bridge latex antibodies with the anti-sperm antibodies. So there would be agglutination, sperms would be trapped, and there could be few sperms that are not having antibodies, they would not link up with these beads, so they would swim freely. So we'll count 200 sperms, and then we'll see percentages, how many of them were attached to the beads or the latex particles, and how many are free. So if 50 less than 50% are bound, then we will say the semen is normal, it is not having the pathological condition, that means they are not having anti-sperm antibodies. And if more than 50% sperms are bound with these latex particle, it means these sperms, they have sufficient number of, or considerable number of anti-sperm antibodies, so they are having anti-sperm antibodies. That was direct sperm mar test. For indirect sperm mar test, we need only the serum or the fluid of the semen, that is fluid of the semen without spermatozoa, from the patient that we are suspecting to have anti-sperm antibodies. So for this procedure, we need diluted fluid from semen, and then we have to incubate it with the sperm of another person that is not having antibodies. So we take semen from another person, from a donor, and we wash its serum away, and we get only the spermatozoa that are not bound with the antibodies, and the semen of the patient and the spermatozoa from the donor, they are reacted, they are incubated together. If serum of the or fluid of the patient has antibodies, then these antibodies they would bind with these spermatozoa of the donors, and then we'll repeat the previous procedure of the MAR test to detect the antibodies. In order to determine antisperm antibodies by using immunobead test, we need polyacrylamide beads that are coated with antisperm antibodies binding antibodies. So these antibodies that are bound with these polyacrylamide beads, they can bind with antisperm antibodies, so they can detect antisperm antibodies. So in case of immunobead test, these antibodies they can directly bind with the antisperm antibodies present on sperm or in seminal plasma. So in case of direct immunobead test, we need the semen that is washed and only spermatozoa are present. So these spermatozoa, if they have antibodies, they are mixed with the polyacrylamide beads that are coated with the anti-sperm antibody binding antibodies. So if antibodies are present on sperms, these beads they will bind with the spermatozoa and make them immobile. So agglutination would occur. So that agglutination would be determined under microscope, and by using same method that 200 spermatozoa would be counted, and then we will see how many percent of them are bound with the anti-sperm antibodies, and then if they are less than 50% that are bound with beads, then we'll say the anti-sperm antibodies are not present in sufficient amount, so semen would be graded as normal. And if more than 50% sperms are bound with these polyacrylamide beads, then we will say the antibodies are sufficiently present, so the semen would be categorized as positive for anti-sperm antibodies. In case of indirect test, as done in MAR test, we need the semen or seminal plasma only, that is the fluid part of the patient that is suspected for having anti-sperm antibodies, and then we need the washed spermatozoa from donor that are not having antibodies, and then we mix them up, and if the patient semen or seminal fluid has anti-sperm antibodies, then these anti-sperm antibodies would bind with the spermatozoa of the donor, and then these sperms they are detected for immunobead test as described previously. So again, if more than 50% of the spermatozoa they bind with the beads, then we would say the antibodies are present in the sperms or semen.